Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Freddie Lawton from Business Review, and I will be your host. It is our pleasure to have Batavia Biosciences with us today, who will be discussing design of experiments in biopharmaceutical development. Today's guest speaker is Danielle Horneman, who is a senior bioprocess scientist at Batavia Biosciences. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you receive via email to rejoin the session. In order to ask questions, you can send them in via the questions widget, so just type them into the box at the top left-hand corner of your screen and click Submit. We allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer in order to provide feedback, and you can do this during or immediately after the webinar. Please use the yellow Help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now please allow me to welcome Danielle.
Hello, good afternoon, everyone. So my name is Danielle Horneman. I'm a project manager and senior bioprocess scientist at Batavia Biosciences. And I like to tell something about design of experiments, how we use that in the development work we do at Batavia Biosciences. So in this presentation, I will first give a short introduction on Batavia Biosciences, so that you know who we are and what we do. And then I go to the basics of design of experiments. And in this part, I like to show you our approach in design of experiments. And what I will do is I will go through the whole workflow of design of experiments from defining the objective to generating the design to, at the end, use the model data that is generated. After that, I will show you one of the DOE examples of the studies we have done at Batavia as far as inactivation process. And of course, I will end with a summary and conclusion of this presentation. So first, something about Batavia Biosciences. Batavia gives support to the biopharmaceutical research and development industry in bringing new or improved medicines to the market. And additionally to that, we also develop technologies in support of global health initiatives. We have facilities at two places, one in Woburn in the United States, where we have Bioseparation facilities, and in Leiden in the Netherlands, where I'm situated. There we also have some laboratories for protein and virus work, and also the possibility to do GMP production up to 200 liters. If you look at the staff at Batavia, you can see that majority of the people working here have a Master of Science degree or a PhD. But next to that, we also have a significant amount of years of experience in industry, which is very helpful in the development work we do. So on the next slide, you see actually the expertise we have. So we have expertise in viral and bacterial vaccines, in recombinant proteins, monoclonal antibodies, and consulting. On the right-hand side, you see the three groups we have. So we have a research group that is looking at clone selection, seed stock, cell line generation. We have development that's working on USP, DSP development, optimization of cell cultures and medium, assay development, etc. And manufacturing, so where we can produce clinical phase 1-2 material, we can do GMP release, stability studies, and give support to our customers. Although those groups are separated on these slides, one of the advantages at Batavia is actually that these groups are overlapping. So the people working in development are also working in manufacturing, which accelerates the transition from development to manufacturing phase. The next slide shows some of our technology and innovation platforms. So we have, for example, STEP technology is a technology that increases protein expression, which leads to faster generation of stable cell clones. We have our SCOUT platform, which is basically integration of our scaled down cell culture systems together with the high throughput purification and assays. and CDIS, which is, covers our experience and complete protocol system on the manufacturing of viral vectors. But we are also working on other technologies and innovations. Last slide I want to show you on Batavia Biosciences is actually showing all the services we provide in research and development, serum tracing and manufacturing, but also our track record on bacterial and viral vaccines and on proteins and antibodies. And as you can see, we have quite an extensive track record already. But now to the main subject of my presentation, in which I want you to tell about design of experiments in process development. And before I do that, I first want to say something about process development. Nowadays, if you work on process development, you should do that according to the quality by design approach. And a definition of this approach is given in the ICH guidelines, which is the development that begins with predefined objectives and emphasizes product and process understanding and process control based on sound science and quality risk management. And the picture below I took from a case study you can find on the internet, which I think represents the idea of quality by design quite well. So you have to start with predefined objectives, which are actually defining the quality of the product you want to have, so the quality attributes. Of course, you'll have a first process design that is based on the knowledge you have, on the product you have. But to further develop this process, you'll have to think about a lot of parameters. So in your process development and process characterization, what you do is you will identify those process parameters that are critical and link those to the quality attributes that are critical for your products. If you have done that, then after your process characterization, you should have an idea what this relation is, and you should be able to define a kind of design space which describes the ranges of all your process parameters, and if you stay within those ranges, you obtain the quality you want to have. And you should have enough information to start with a draft control strategy. So how do you make sure that your process parameters stay within the design space so that you will guarantee that you have the quality you want to have? When you have done that, of course, you go further in verification of your process test if it's indeed robust, and then you end up in your production cycle and you continuously check your performance of your process, update your control strategy. So that is the idea of quality by design, that you first define all the attributes, quality attributes, and link your process parameters to those attributes to make sure that in the end you can just guarantee you have the quality you want to have. Well, how do you get that? So there are several tools to come to your final process. Of course, if you do process development and process characterization, you will do experimental work where you identify the process parameters that are critical and you link these process parameters to your critical quality attributes. But also, as the definition of quality by design says, you should use quality risk management throughout your whole process development. So in the beginning, that can be that you use risk assessment to select those parameters you want to take in your process development or later on in your process characterization. But also for your control strategy, you will use risk assessment to see which parameters do you need to control to guarantee your product quality. Of course, in this presentation, I will talk more about how you can perform the experimental work. So basically, you have several tools to do your development, like I said. So your risk assessment to identify the critical quality and performance attributes. Of course, quality is very important, but we all know that also the performance should be good enough, as you want to have a low cost of goods. So you want to have high yields, for example. Experimentally tools are, for example, high throughput technologies and strategies like we have Scout, but also design of experiments that can help you in accelerating your development to get faster to the answers you want to have. And why is that? So on the next slide, if you look at experimental work, you can define three main objectives why you would do experimental work. First, you want can be a screening in the beginning when you don't know anything about your process and you just want to know which parameters do influence certain attributes. That's more screening, just try out some things. If you're a little bit further in development, you want to know, okay, but where do I have to be exactly to be in my optimal point? So that's more an optimization objective. If you have defined your process, then you want to control your process, but you also want to show that your process is robust. So then you want to do some experiments showing you have a robust process. Well, a challenge in process development is that with all those steps you have and all those process parameters, you have a lot of factors and responses that you can study. If you look in a traditional method that is depicted in the figure on the right side of the slide, you select first one factor, for example, factor number 2. You perform some experiments represented by the black dots in this figure, and you find that in the light orange part you have your optimal point for factor 2. Then you say, let's change another factor, for example, factor 1. You change that around this point, and you find an optimal point in the more dark orange area. But in fact, your optimal point is in the red area, and with those single factor experiments, you will never find this. And that's why design of experiment is so important, as that is a mathematical method that you can use that can help you for planning and conducting your studies in a systematic way, and it actually manipulates multiple factors at the same time to determine the effect of all these factors together. So instead of performing experiments around and only changing one factor at a time, you can do with less experiments where you vary multiple factors, as indicated now in the figure on the right. You see 4 black dots representing 4 experiments. You can already get so much information that you know in which direction your optimal area can be found. So design of experiments, you can do less experiments but get much more information out of it. Okay, how should we approach design of experiments? I will now go through the workflow of design of experiments. Of course, like I also said in quality by design, you always have to start with defining your objective. What do you want to do? So if you are at the early stage of your development, you probably have a lot of parameters you want to investigate. Well, the first thing you can do then is do a screening, identify the factors that influence the different attributes. And you can get already some information about ranges of these factors. If you're a little bit further in process development and you know the factors that do have influence on your attributes, then you want to do maybe some more experiments where you can find the relation between the parameters and these attributes, your responses, so you can get a model that you can use for prediction and for finding an optimal point. And once you have your process developed, you want to show robustness. All those experimental objectives require a different approach in your design. Well, if you have your objective, of course you have to think about which responses do I want to study. So which attributes are important for the experiments I want to do. So that can be either quality attributes related, so things like purity, or performance attribute related responses like yields. Then of course you have to select the factors you want to take into your design. And to determine the factors you want to include, you can, of course, use some risk assessment tools. You can use your prior knowledge, look in literature, know what is your experience in previous processes you have developed, and in that way select the factors that might have an effect on the response which you want to study. Another thing to think about if you define factors is the type of factors. You can have quantitative factors, and I think most of them will be quantitative if you want to check different concentrations of additives or conductivity or temperature. That are all quantitative factors. But you can have also qualitative factors. For example, if you do a medium selection, that is not always easy to quantify a medium, so you can just say medium A, B, and C and take that as a factor. Also important to realize is that in your design you of course will take the controllable factors, so the factors you can easily change value of, so you can set the conductivity at 10 millisiemens or at 20 millisiemens, but you might also have uncontrollable factors that do influence, do have an effect on the response. Maybe the humidity of the room or the temperature of the room cannot be controlled, but might influence the outcome. You cannot really put those in the design, but you can think about it of monitoring those factors, so that at least if you do the analysis, you can take those into account. Well, if you have your objective, you know what you want to look at, which responses, you know which factors you want to investigate, you're ready to generate a design. And in the generation of design, you have to make a selection in the type of design you want to use. You have to think about the model you want to use after you have produced your data, because you have to think about what this model can tell you about the data. You have to think about the ranges of the factor. So what are the values you want to put in your design? The order of the experimental runs you're going to do. Blocking of factors, and I will talk about that later. Do you want to include center points? Are you going to replicate some of the runs? All those things you have to think about when you generate your design. The next few slides will therefore go about, tell you more about generating a design. So if you look at the next slide, if you want to select a design, you'll have to think again about, okay, what was the objective I want to have? Because each design, not every design is suitable, for example, for optimization objectives. There are a lot of different designs, but in this slide I show you the two basic designs to explain how design looks like and how the models are related to the designs. So on the first row you see the designs that you can use for screening studies or robustness studies. So for screening studies you want to find the factors that influence responses, and in robustness studies you actually want to show that the factors in the ranges you selected have no influence on the response anymore. Different objectives, but the approach is basically the same. So in the second column you see design itself. On the left hand side you see a design for two factors, where the black dots represent again the experimental runs, and they are located in a square area. And basically that's, if you look at the corner point on in the left, the bottom left, that's actually the run where both factors x1 and x2 have a low value. If you go up, you only increase the value of your second factor x2, whereas the value of the first factor stays low, and in the corner point on the right hand side on top, both factors have a high value, and the other black points on the right-hand side on the bottom indicates a high value for factor 1 and a low factor, or for a low value for, high value for, factor 1 and a low value for factor 2. You can also make the same representation. If you have 3 factors, then you go to a more cubic-like representation. But of course, you can do this kind of designs where you select a low and a high value for your factors if you have 4 factors or 5 factors, 7 factors, 8 factors. The number of runs, so the number of black dots you see, can be calculated by 2 to the power of the number of factors, k. So k stands for the number of factors. You see in the figures also a white dot that are the center points, and I will come back later to that. On the the right column, you see the models that you can use. So that are the models that can describe the outcome of these designs. And the first one is a linear. So that is a model that takes into account the effects of each separate factor. And you have an interaction model that is basically the same as a linear model, but added to that are the effects of the factors combined. So you see here the beta 1, 2 times x1 and x2. So the beta 1, 2 is the coefficient that describes what the effect of the combined factors is on the response y. A drawback of these kind of models is that if you change one factor from a low to a high value, you can only describe that with a straight line, a linear approach that is. So if that is not the real representation of the real world, because it should be a curved line, this model is not suitable. And for most optimization studies, you might need curved lines. And that is why for optimization studies and characterization studies, you need another type of design where a third level is included. And an example is given below with the composite designs. So they're basically the same as the factorial design. So you have the corner points, but you see that 4 additional experiments are added in a 2-factor designs on the factor axis, creating a third level for each factor. So the disadvantage, of course, is that the number of runs is increasing with 2 times the number of factors. But the advantage is that you can also use quadratic models to describe the outcome of these designs. And that is beneficial if you want to search for an optimal point. Well, these 2 designs you see here are I think the basic designs in design of experiments. But everyone who has read about design of experiments probably knows there are a lot of other designs possible. So on the next slide, I just mentioned a few of them. So I just talked about factorial designs, but you also have fractional factorial designs, where you see that not all corner points are taken into account in experiments. And it is done to reduce the number of runs in your design of experiments. And that for 3 factors, that might not be an issue, because you don't have 8 experiments. But you can imagine if you have 7 factors you want to investigate, you add up with 128 experiments, and that might be a little bit too much. So you can then decide to leave out some of the experimental runs and still get information about effects. The only thing you have to take into account that some of the effects will be confounded. So you cannot always distinguish between each separate effect. But if you think in advance about what factors you think will affect the most, you can make reasonable selections in the runs you want to do. On the right hand side you see another example of a Box-Benken design, which is 3-level design, so can be used for optimization. It's a little bit less powerful, less experiments than a composite design, but you can maybe think of experimental setups where you cannot perform the experiments in the corner. So this might be then an alternative for a composite design. What is also often used is de-optimal designs, and that are because in a lot of occasions it is not possible to do experiments on each corner of your experimental design area. So in what you see in this picture, I had a red triangle in this square, and in this area, this red area, it's not possible to perform experiments because maybe your equipment is not capable of doing it, or there are other restrictions why you cannot come into that area. So you have to think of other design points, but still you want to have a powerful design that gives you the answers you want to have. So there are algorithms, and I'm not going to explain you that, but there are algorithms that can help you in defining the design points you want to have. And as you see, that doesn't have to be symmetrical anymore like you've seen in the other designs. So that's often used if you have irregular experimental regions, but sometimes you already have a data set and you just want to extend, and then the optimal designs can also be very helpful. And of course, there are many other designs for specific situations you can use. It's too much to talk about it right now. So we have now talked about the type of design and the model selection. Now have to think about the ranges we want to select of the factors we selected for the design. Of course, the ranges are depending on what is feasible in your experimental setup, but also don't forget to think about the feasibility you have in your process. It doesn't make sense to test in between 20 degrees Celsius and 500 degrees Celsius if you know that your process will never be able to go to temperatures higher than 50 degrees Celsius. So also always think also about the process you want to go to. Of course, also the objective of your experiment is important. For screening, you want to choose maybe somewhat wider ranges because you want to see if there's influence of your factors. Whereas for robustness, you will often choose smaller ranges because you want to show that within those ranges your process is robust, so there is no effect of those factors. You also should think about the variability in your experiments and your analytics. And to show you that, you can look at the graphs that now appear in the slides. So if you have high variability and you select very small ranges, you will be able to draw a line through the data points. So you will have a model that describes the data points. But if you look at the confidence intervals of those data points, you will see that the confidence interval of the slope of your line is also very broad. So you have a poorly defined slope of your model line. Whereas if you take a wider range of your factors, you will be much much more able to predict the correct slope of your model line. So think about it when you select the ranges of your factors. Another thing, I briefly mentioned that before, are the center points. So it's very normal to include some center points as a reference point. And if you look, for example, for two-level factorial designs, as I explained a few slides before, if you have only 2 points, a low and a high value, you can of course see what the effect of the change of that factor is on the response, as you can see in the figure next to the text. But the only thing you can do is draw a straight line between the 2 points. Whereas maybe in reality, it's not a straight line. Well, if it is a straight line and you have added center points to your design, the center points should fall on this straight line. Well, in this case, in this example I show here, you see the center points are lying far below the line. So the only way to have a model that is describing all 3 points is by having a curved line. So even if you do a factorial design, by adding center points, you can at least already get some information about curvature. Is there curvature? So if you need to do other experiments later on, you can see if you do need another type of design to optimize your process or not. It's also important to take some replicates into your design, and often that is your center point. That's your reference point, so you take that a few times in your design. And the reason to take that into your design is in the end you will get a model describing your data, and your model, of course you want to fit it perfectly, but it will never fit perfectly, but you want to know what the reason is for this not perfect fit. So is that because the model is not correct, or is that just because I have some error in my experiments, some variation in my experiments which he cannot model? But to be able to define or determine this lack of fit, you need to include some replicates. And as your center points often is a reference point with data that has average value, so it's not on the high side of the response or low side, that's often the best point to take along and take some replicates in your design. Okay, so now talked also about center points and replicates. Then one thing about the order of experiments. You could, of course, just do the experiments and increase first one factor and then increase the other factor. But then you'll have a risk that if there is an other factor that is not controlled in your design, that is maybe time related, maybe the temperature is increasing during the day, that might be then by the model be related to one of the factors, which is in reality not the case. So to prevent unwanted variability in your measured data, it's better to randomize the order of runs. So systematic effects will be transformed in random noise, but it also helps you later, if you analyze the data, you can actually see if there is some time-related factor you did not control in your experiments, and you can think about that on which one that can be, and maybe take that along in your next design. However, sometimes it is not possible to randomize all your factors, or maybe you cannot do all your runs in one series. Maybe you have a batch size that is not large enough to perform all your runs, so you have to produce a new batch, which might be different than the first batch for your second, for the other runs. In those occasions, you can block your experiment. So that means you define several blocks in which you do your experiments, and the block itself becomes a qualitative factor. So an example is given below. So you have block, for example, a batch size that's only sufficient for 6 runs. So you can see in first block you perform all corner points and some center points. Then you make a new batch, and then you perform the other points of your composite designs plus some extra center points. And these block factors will be taken in your analysis as a qualitative factor, so you can see if the block itself has an influence of the outcome of your response. So now we have generated the whole design, and then you have to do your experiments. As I said, that should be done in the randomized order. And often design of experiments is done in a scaled-down setup. Because you cannot always do all your experiments on a process scale. That's fine, you can do that. But always, if you really want to use the data later on in your control strategy or process characterization, you always have to show later on also to the authorities that the scaled down model you have used is representative for your large scale process. So if you scale down your setup, check if your setup is indeed representative for your large scale process. Otherwise, you might get into problems later. Then you have performed the experiment, and then you have evaluate your data. And of course, everyone, you want to model the data. You want to describe your data with a model. But it's good just first to look at your data. How is your data looking? So one thing you can look is at the replication runs. Are they giving the same result? So is the variation in the replication runs, is that small compared to the variation you see in the response of the entire data sets? If you see a large fluctuation in your replication runs, you might rethink about your experimental setup, and you probably will not find any relation between your factors and your response. Another thing to look at your data set is just see how the data is distributed. For a good analysis, it is desirable to have a normal distribution of your data. So if you see tailing, like you see in the figure below, there you see the count of responses between certain values. So you see that we count 5 responses with a value between 1 to 5, 3 responses with a value between 5 and 9, 2 between 9 and 13, and then some responses with higher values where we only count 1. So you clearly see a tailing in this data set. That normally indicates that you better can transform your response. For example, by using a log transformation, but it can also be other transformation as well. And in this case, if we perform a log transformation and then count against the log of the response, we see that the data set we get is looking more like a normal distribution, and this is a much healthier data set to analyze. So let's go to the data analysis. What is used for data analysis is analysis of variances. But of course, there are also several diagnostics tools to see how well your model is or if there are outliers, etc. I will not go in explaining you exactly how all those statistical tests are working or are being performed. I think the most, if you're going to use with design of experience, you probably will use some software packages. There are several software packages you can use. But the next slide I will briefly explain on the analysis. So to give you a little bit basics on how these analyses work and what you can do with it, what the answers tell you. So first, analysis of variance. That's basically based on F-test, and an F-test compares the variance in 2 data sets and compares the variance to see if the 2 data sets are from the same group or from different groups. So in that way, you can test if a change is changes you see is affected by a certain factor, or that it is just the same, it is within the variance of the whole data set you see. So the F-test, what it does, what it gives you is a probability that the defined hypothesis is true. And in DOE you have basically 2 F-tests you can perform. One is a hypothesis that you say that the model or a model term in the model has no significant effect on your response. And of course you want to get a significant model, and you only want to have significant model terms. So the desired value, as is given in the table on the right hand side, is that the probability that this hypothesis is true should be very low, because you want the model to be significant. The second test determines the significance of lack of fit. So what the hypothesis there is that the model error equals the replication error. That's actually also what you want to have. So here you want to have a high probability. Because if that's not true, then that means that your model is not correct. So you have to maybe change your model. So that one you want to have a high value, so above 10%, so above 0.10. Then of course you want to look at your fit. The goodness of fit, I think everyone knows that if you have several points and you do a linear fit through it, you get a goodness of fit, the R squared. Well, if you have multiple factors, well, if you put a lot of factors in your model, you will have a high goodness of fit. So you should adjust that for the number of factors you have in your model, and that's the R squared adjusted. But you can also look at the goodness of prediction, and that says something about, okay, if we have a new data point, how well can we predict that? Those should be at least 0.6. I know for linear regression you always look for 0.9099, but if you have multiple factors, sometimes you can be already satisfied with 0.6. Well, what do? You select, if you have your software, you select the significant model with the best fitting model. You look at the model terms, remove those that are not significant, and then you check with, you have several diagnostic tools to check for outliers, for trends that you didn't expect, or which data have a high influence on the model. And I want to say a little bit on the diagnostics. I'm not going to show all kind of diagnostics tools. Every software package has a lot of those tools you can use, but it's good to go through them after you have selected your model. One that is certainly will be there is a normal plot of your residuals, and your residuals should be distributed in a normal way. That should give a straight line in your plot. And in this case, so every dot in this left figure represents one of the experimental runs. You see one dot is almost in the right top corner, so that might be an outlier. So that is a data point you should investigate. You can look at the residuals versus runs, so to see if there are any time-related variables, because this residuals versus run plot should be very random around the zero line. You should not see any trending there. And you have a lot of diagnostics that look on the influence of each data point on the model. And one example is the leverage versus run, and the leverage looks at what the influence is of that data point on the model. And the higher the value is, the more the model is forced to go through the point. So if the value is 1, that actually means that the model was forced through that point. So if you take that point out, your model will change significantly. Having said this, so looking at the diagnostic, if you see a point like this in the right-hand figure, so this red point with a value of 1 for the leverage, it doesn't mean that you can remove this point. So don't remove the points from your data set just because the diagnostics indicators might be a problem. The value might be right, but it is worthwhile to investigate those data points. It can also indicate that you have not selected the best model, or maybe your experimental setup was not the best. So maybe the ranges you selected should have been smaller, anything like that. But always look at the diagnostics and only remove it if you're sure that the data point there is wrong. And of course, last slide about this part is use of the model. Well, if you have done some screening experiments, you can get a model, you can get all the coefficients of the factor describing the effect of that factor. You see in this picture the coefficients for the several factors, factor 1, 2, and 3. And then AB represents the coefficient for the combined effect of factor 1 and 2. And you see the difference in the coefficients, and you actually see, for example, that the combination of factor 1 and factor 3, well, you can't even see it in this graph. So if you go further in your study, you don't have to take those interactions along in your design anymore. And even factor 3 might be can be ignored in the following studies you can do. So maybe you can only continue with factor 1 and 2. If you have done optimization studies, you want to use your model for prediction or optimization of your process, define design space, operating space, think about your control strategy. So you often see there are those kind of response plots you see here. So you have a response plot for factor 1 and factor 2, and the different colors in this plot represent different values of the response. So blue is low values and red is higher values. And from this response plot you can define the area where you have to be to reach your quality. For example, if your response needs to be higher than 30, you can define the area where you factor 1 and factor 2 should be to obtain this response. Please take into account that this model, of course, also has this confidence interval for each coefficient in the model. So that are the two different colors you see in the right-hand figure. So the orange area gives a high probability that if you are in this area, you will stay, you will have a response of higher than 30, whereas the yellow is just outside the 95% confidence interval. As I said, when scaling down your experiment, also, of course, also when you have your model, you should be aware that you have to check and verify that this model is indeed describing your production scale process. I see my time is going quite fast, but I still want to use the last minutes to show you one example of a DOE study we performed at Batavia. And it is one of inactivation of a viral vaccine. So inactivated viral vaccines, when those are produced, somewhere during the purification process, there is an inactivation step where you inactivate the virus. And that can be done by chemical addition of BPL formaldehyde, that are most common used, or physically by heat, gamma irradiation, UV treatment. But you can imagine if your inactivation is not working fine, that has severe effects on global health. So you really need to understand your inactivation kinetics. That is a requirement on your inactivation process. You really need to understand the kinetics, and you also should show that you can reproduce the same kinetics every time again. And in the end, of course, you also should show the completeness of your inactivation. So on the bottom, you see some examples of inactivated viral vaccines. Well, we had for a project done a lot of process developments in which also inactivation was a step. Well, you might recognize that you have done so many runs. In the end, we had done more than 70 inactivation runs. But then we put all the data together, and then we actually saw quite some variation in the kinetics and also in the yields we did not expect. And then we looked again at all those runs, and we saw that we actually changed a lot of factors over time during development. So factors in conditions, in equipment settings, skills, etc. And it was not clear to us which factors caused the variation. We could define several processes where the combinations of all the factors were the same, but between the different processes, multiple factors were changed, so we did not know which factor really caused the variation. So we needed to investigate that because we need to understand the kinetics of our process, of our inactivation process. And we thought, well, if we can explain what we see, we can also maybe use that model for further optimization and defining process control. So the workflow of this experimental work, of course, this is, if you look at objective, is more optimization and characterization. We selected factors based on our risk assessment, on the knowledge, looking at all the previous runs, and we selected 6 factors, all quantitative and controllable. Of course, selected responses, for example, the inactivation rate and the yield, and we generated design. First, we did a small screening experimental run to reduce number of factors to 4, and then we did a central composite design. Defined the ranges based on the ranges used in the past and our knowledge on the variability, and of course we added multiple center points. We scaled down the inactivation, but we really had to scale down it because the material was limited. But we checked in the first screening, we checked the reference process that it was the same as the process on the large scales we were running, and that was the case. So we knew that our small scale did describe the large scale. Then we generated the data, and what I said, you should look at the replicates. Well, you see here an example. The blue squares are the replicates, and the red dots are all the other experiments. And you see that there is there's a very small variance in the data of the replicates, whereas the variance of all the other data is much larger. So this is a good data set to work with. And also, the distribution of the data looks very nice. Then we put a model, so we used the quadratic model, we removed the non-significant terms, we did the diagnostics. Sorry, I go a little bit fast through the slides, but this I want to show you because we had a quite nice model, but when we did the diagnostics, there were a few of those diagnostics tools like the normal plot, but also residuals versus predicted, that every time one of the runs was very different from the other runs. And for example, in the Cook's distance with the right-hand figure, you see it has a very high value, and the Cook's distance says something about the regression of your model. If you take this point out, the regression will change significantly, so you will get a different model. So we checked this data, and we actually found that the inactivation rate was based on invalid data, so we were actually not allowed to use it. It was completely wrong. So we removed this data from the data set and then did the analysis again. And then we find a very nice significant model, as you can see in the table. And the lack of fits also very nice. So the model really described was really significant. And if you looked at the R-squared, the goodness of fit was very high. And you can also see that in the figure on the right-hand side, where you have the predicted versus actual, and you see that is they're all on the line. So we could very well predict our experiments we have done in the design. Of course, more important is, could we predict with this model the variance we had seen before? And that's on the next slide. And you can see that we actually pretty well could predict the outcome of the runs we did in the past with this model. So you see the red diamonds are also very close to this line where predicted versus actual is the same. So we were very happy with these results. And of course we had now a model that could describe the process. That means we could also use that for optimization, and we also had some first input for control strategies, so which factors are important to control. So my last slides, just a summary of the presentation. Well, from the inactivation example, I hope I could show you that with only 2 experimental runs, so we did a small screening and after that a composite design experimental run, we tested more than 40 conditions in a very short period of time, and we got a model that explains the variation we had observed before. We identified the factors that influence the rate and the yield, and this is very important for further optimization and defining your control strategy. And So I also hope with this presentation that you've seen that design of experiment is really useful tool in quality by design, and it can be used from the early stage development where you do screening up to the end where you can do robustness testing, and it helps you in acceleration in the process development and characterization. So with this I come to the end of my presentation. I see I took quite some time. I hope you don't mind, but but we probably have some questions, and I know Freddie collected all the questions, so I want to ask Freddie to help me with that.
Perfect. Thank you very much, Danielle. So just a quick reminder for the audience, in order to ask questions, you can send them in via the questions widget. So just type them into the box at the top left-hand corner of your screen and click submit. So let's have a look at the questions which have been submitted already. Danielle, here's one for you. Someone says, on what kind of projects does Batavia use DOE?
Yeah, really several projects. So we have really in the beginning of the process development. So for example, where we have to do medium selection for USP process, we use DOE a lot for defining optimal conditions in chromatography steps in the purification. Indeed also for inactivation, for if you USP process development, so looking at different additives added to the bioreactor, or things like stir speeds, etc. So really throughout the whole process chain we use design of experiments, yeah, from the beginning of the development up to the end.
Great, thanks Danielle. Next question, someone says, what is, from your experience, the maximum amount of factors you can use in a model?
Yes, there's not one answer I can give to that, because that's it also depends on the experimental setup, what you can do, and the material, of course, you can use. When people come to me and ask me for help in the design of experiments, they always come with a really very big list of factors to include, and most of the time they also directly want to have an optimization of the process. Well, I will never advise you to do an optimization with more than 6 factors, because that you get really a lot of experimental work to do then. So in the screening you can select more factors. You can, yeah, what I said, depending on how your experiment look like, how easy it is. Sometimes you can do 100 experiments. If you have a high throughput system, you can do a lot of experiments, so you can add 10, 12 factors to it. But if your experimental setup is somewhat more difficult, I would limit the number of factors in your design to maybe 6 or 10. And if you go to optimization, I really would advise you first do some screening to reduce the number of factors you're going to use. So it's really depending on the feasibility in your experiments, what you can do. If it's easy to do 100 experiments, you can really add a lot of factors. But I really advise everyone to do a risk assessment and really think about, is it really necessary to take this factor along, and do I expect interactions of combination of factors, because then you can also reduce the number of experiments. And so really think about, what do you expect from these factors. I hope this answers a bit the question.
Thanks, Danielle. Very clear answer there. Let's see what else we've got. So one audience member says, do you need to be statistically educated in order to use DOE?
I think you should have a basic understanding of statistics. So, like in this presentation, I didn't go into detail how you can, how can you calculate the probability using an F-test. I mean, normally you have software packages to do that. So that is not really necessary. You don't need to know all the equations. You have software packages enough that can do that for you. But it will generate an answer, and you need to know what this answer means. And I hope in this presentation I have explains a little bit, for example, with the F-test, what the answer means. And also if you design your experiment, so you need some basic understanding of statistics, but you don't need to be able to perform the statistics yourself, but at least you should know if an F-test is done, you should know what the answer means. Otherwise, it will be very difficult to use the results, or you can maybe misinterpret the results.
Great, thanks a lot, Danielle. That's about all we have time for, I believe. Don't worry if your question wasn't answered during the session. It's possible it was perhaps a bit too specific for Danielle to answer it now, but she'll aim to get back to you personally via email. Danielle, is there anything you'd like to add before we conclude?
No, I just want to thank you that I had the opportunity to give this presentation, and of course thank you for everyone who listened to this presentation. And that's it. So thank you.
Great. So that just leaves me to thank Danielle for what was a great presentation, and to thank Batavia Biosciences for sponsoring this session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. Once this webinar ends, today's survey will appear in its place, and we would appreciate it if you answer the questions in there. And we look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you once again, and I hope you all have a nice day.
Thank you. Bye.